Good morning, friends. Myself, Abdullah Sharikar, Assistant Professor, Department of Pharmacology, SVKM Institute of Pharmacy, Dubai. Today, we are going to study about the cell part 3. Earlier, we have seen cell part 1, cell part 2. Now, we are going to study cell part 3. After completion of this lecture, the learners will able to understand the structure of cell organelles, functions of cell organelles, and some question banks. Now, the first is the ribosomes. Ribosomes are considered as the site for the protein synthesis. They contains high level of ribonucleic acid, that is the ribosomal rRNA. They contains more than 50 proteins, and ribosome consists of two subunits, that is the small and large subunit. These subunits made separately, particularly in the nucleolus, that is inside the nucleus, and they exit separately and unite in the cytoplasm. These subunits, particularly there are some subunits which are attached to the plasma membrane, or particularly to the endoplasmic reticulum, and there are some subunits which are free in nature. The free ribosomes synthesize the proteins, and there are some ribosomes which are also localized into the mitochondria, where they synthesize the mitochondrial proteins. As you can see here, the structure of the ribosomes, the upper larger part is called as the larger subunit, and smaller part is called as the small unit of the ribosome. Second important organelle is the endoplasmic reticulum. These are the membrane organelles, and particularly they extend from the nuclear envelope. They are originated from the nuclear envelope. Envelope is nothing but the outer covering of the nucleus, and the endoplasmic reticulum are considered as the flat sac or the tubules like a structure. And there are two types of endoplasmic reticulum. One is the smooth endoplasmic reticulum, other one is the rough endoplasmic reticulum. As I already told that the endoplasmic reticulums are considered as the membrane network. Now, first we consider the rough endoplasmic reticulum. It continues with the nuclear membrane. It is covered with the ribosomes. It is very essential. Because of this presence of ribosomes, they act as a site for the protein synthesis. That is why the rough endoplasmic reticulum are called as granular endoplasmic reticulum. Second point, they are also essential for the plays an vital role or the critical role in the synthesis of the glycoproteins and phospholipids. Already I told that they are considered as a site for the protein synthesis. That is why they are granular in nature. Now, the next is the smooth endoplasmic reticulum. They are extended from the rough endoplasmic reticulum. Please pay attention here to differentiate between this smooth and rough endoplasmic reticulum. Rough endoplasmic reticulum, I already told that they are extended from the nuclear envelope, whereas the smooth endoplasmic reticulum, they are extended from the rough endoplasmic reticulum. Second important point is that the smooth endoplasmic reticulum plays an important role in the smooth muscle contraction. When the contraction process is occurring, that time the sarcoplasmic reticulum, that is the one of the important form of the smooth endoplasmic reticulum, it releases the calcium ion, and which plays an important role in the smooth endoplasmic reticulum, whereas the rough endoplasmic reticulum fails to show or fails to play an important role in the contraction. The smooth endoplasmic reticulum does not contain ribosomes. That is why there is no protein synthesis, and therefore, the smooth endoplasmic reticulum are agranular in nature, whereas rough endoplasmic reticulum contains ribosomes. So, there is a synthesis of protein, and that is why rough endoplasmic reticulum are granular, whereas smooth endoplasmic reticulum are granular in nature. Next point, in liver or cells or the kidney cells or the intestinal cells, the smooth endoplasmic reticulum causes the liberation or the release of the phosphate group from the glucose 6 phosphate, and that is why the smooth endoplasmic reticulum directly releases the glucose in the blood stream. Next point, the smooth endoplasmic reticulum plays an important role in the synthesis of fatty acids and steroids, say example estrogen and testosterone, and particularly more important that liver cells, particularly the smooth endoplasmic reticulum present in the liver cells, they plays an important role in the detoxification of lipid soluble drugs or the harmful substances, say example alcohol, say example pesticides, or say example carcinogens. Carcinogens are nothing but the cancer producing drugs. These drugs are detoxified or made harmless by the smooth endoplasmic reticulum. Now, there is a relation between the drug tolerance and the role of smooth endoplasmic reticulum. As already told that the function of smooth endoplasmic reticulum is to detoxify certain drugs, that is the lipid soluble drugs. The individuals who repeatedly take sedative drugs, particularly sedatives are nothing but the drugs which calms the patient or the individuals, such as example is the phenobarbital. These drugs have tendency to increase the amount of the smooth endoplasmic reticulum and its enzyme in the liver. So, after prolonged administration of these drugs, there is a development of tolerance, means there is need to take higher dose of drugs to produce original effect, and in that case due to increased dose there is a development of drug dependence, and that is why the patient has to take larger amount of drug than usual. Third important is organelles is the Golgi complex. The Golgi complex, the first synthesized proteins are transported to the Golgi complex, means from the endoplasmic reticulum, particularly rough endoplasmic reticulum, the proteins are transported to the Golgi complex. The Golgi complex contains cisternae. What are the cisternae? These are the 3 to 20 small and flat like structure with bulging edge, and the bulging edges have the curve in nature or the curve shape. That is why the curve shape of cisternae gives the cup like structure to the Golgi complex. It has two two surfaces, one is the convex and other one is the concave. The convex, that is called as the entry of the entry point to the proteins molecule. This convex entry or the cis face of the cisternae or the Golgi complex oriented towards the rough endoplasmic reticulum. As already I told that the after protein synthesis into the rough endoplasmic reticulum, they are first transported to this Golgi complex through this convex entry or through this cis phase, whereas the synthesized the entered proteins into the Golgi complex, they are exit or they are leave the Golgi complex through its concave exit, that is trans phase, and it is oriented towards the plasma membrane. The space between this cis and trans cistern is called as the cisternae, as you can see in the picture. Now, what are the functions of Golgi complex? More importantly, Golgi complex plays an important role in the modification, sorting, packaging and transportation of the proteins received from the rough endoplasmic reticulum. Means here the proteins which are coming from the rough endoplasmic reticulum, they are modified, they are sorted, they are packaged and they are transported to the next organelles. Second important function of Golgi complex is they form the transport vesicles, means whatever the proteins are present or the came into the uh, Golgi complex, they are modified. After modification, they are transported to the other molecules or the other cell organelles, say example lysosomes. That is why Golgi complex plays an important role in the transportation, synthesis of transported vesicles for the proteins. Next, more importantly, they also plays an important role in the secretory formation of secretory vesicles and discharge of the proteins, particularly into the extracellular fluid via exocytosis process. And last, more important function of Golgi complex, they plays an important role in the formation of membrane vesicles that transport the new molecules to the plasma membrane. Means the Golgi complex plays an important role in the modification, sorting, packaging and transportation of the proteins with the help of formation of transport vesicle, with the help of formation of secretory vesicles, with the help of formation of membrane vesicles. This is the structure. You can see there is a medial cisterna, entry phase cisterna and exit phase cisterna. Next important cell organelles is the lysosomes. Lysosomes have diameter 250 to 750 nanometer. These are the membrane enclosed vesicles and they are formed from Golgi complex. The lysosome contains near about 40 to 60 digestive enzymes, particularly hydrolytic enzymes in the form of hydrolase. They work at acidic pH and they contains H plus ion pumps. It is very important. Lysosomal anterior has pH 5 and its side outside, that is the cytosol, has pH 7, which is 100 times more acidic in nature. And the lysosome contains transporters which carry the transportation of the glucose, fatty acids and amino acids into the cytosol. Functions. First function is autophagy. Autophagy is nothing but the engulfing, digesting of the organelles, and the digested organelles are again reused, that is reuse of the digested organelles. These mechanism, that is the engulfing, digestion and a reuse mechanism, that is called as the autophagy. And that is why if you consider regarding the liver cells recycle its half cytoplasmic content every week. Means every week half of the cytoplasmic content of the liver cell is engulfed, digested and reused. And they also plays an important role in the cellular differentiation, means identification of the cells, growth of the cells, tissue remodeling and cell defense. Again there are some functions. Second function is the autolysis. Autolysis is nothing but the destruction of the entire cells. Here it is very important to note that the lysosomal function, that is autolysis, is very essential, particularly in the pathophysiology of certain diseases. And more importantly, after death the tissue is deteriorated or the digested with the help of autolysis, degraded with the help of autolysis. There are some extracellular lysosomals. They plays an important role in the fertilization, particularly penetration of the sperm during the cyst. And this is the structure of the lysosome, where you can see the hydrolytic enzyme and transporter proteins with membrane. One important disease which is related to the lysosome, that is the Tay-Sachs disease. It is caused by the faulty or absence of lysosomal enzyme. Particularly the prevalence or the cause of this Tay-Sachs disease you will observe in the children of Ashkenazi, that is Eastern European Jewish. Why it is happening? Why it is occurring? Due to the, it is an inherited condition characterized by the absence of single lysosomal enzyme. Single lysosomal enzyme, that is called as the HEX capital A. Due to the inheritation of the HEX A, the Tay-Sachs disease occur. What is the role of this HEX A? This enzyme break down a membrane glycolipid called as the ganglioside GM2. This HEX A enzyme plays an important role in the breakdown of ganglioside GM2, which is more abundant in the, or the, which is present in higher concentration in the nerve cells. So, as there is excess of ganglioside GM2 accumulation occur, and the nerve, due to this higher ganglioside GM2 accumulation, the nerve cell function less efficiently, means the nerve cell become function of the nerve cell deteriorated, and in that case the children's or the patient experience seizures and muscle rigidity, and they gradually become blind, demented, uncoordinated, and die before the age of 5. This is the question bank. Write the structure and function of ribosomes, write the structure and function of endoplasmic reticulum, differentiate between rough and smooth endoplasmic reticulum, write structure and function of Golgi complex, write structure of and functions of lysosome. And these are the references I have taken from Guyton Hall medical textbook of medical physiology and principles of anatomy and physiology by Gerard Tortora. Thank you.